All right, welcome to our next lesson on Unit 6, which is DNA replication. And if you don't get this meme now, hopefully by the end of the video you will. Trust me, to biologists, it's hilarious. All right, so before we get into how DNA is replicated, let's remember a few things about DNA structure. So DNA has a specific directionality. Just like when you're reading or writing in English, you write and read from left to right. DNA also has specific ends. So one end is called the 5' end. And the 5', that apostrophe right there is read as prime. That 5' end has a free phosphate group. So that is a phosphate right there. And what you can do to try and remember this is to remember that the Ph that starts out 5 is the Ph that starts out phosphate, and link those two in your mind. The other end is called the 3' end. It has a free hydroxyl or OH group that comes off of the 5 carbon sugar, in this case a deoxyribose. And the important part of this is DNA is synthesized in the 5' to 3' direction always. And this is going to become very important for one of the kind of more complicated concepts for DNA replication. So notice there's an arrow going from 5' to 3' because that's the direction in which DNA is replicated. DNA replication as a process is called semi-conservative because what happens is the original DNA molecule separates into the two complementary strands, and each strand of DNA is going to serve as a template or a guide to produce the new strand. So once you're done with this, at the end of the process, you have two DNA molecules formed, each of which has one original strand from the original molecule, which here is represented in blue on this diagram, and then one newly synthesized strand here represented in red. So the DNA is half or semi-conserved. This is really useful because later on, if you're proofreading after you've replicated your DNA, if you have some sort of marker to show that this is the original strand, if you find a mismatch or a mistake in your replicated DNA, a base doesn't quite match, you know which one you need to fix. You don't fix the original strand, you would fix the newly synthesized strand. So this does help to allow the cell to correct some of the mutations that might happen in the process of DNA replication. So remember our base pairing rules from talking about the structure of DNA. Because of this, DNA strands are complementary to each other, meaning they match. So you can use the sequence of one strand to predict the sequence of the opposite strand. So if we remember, A pairs with T, C pairs with G, T pairs with A, and then we can just continue predicting the strand that is complementary to this strand. And that is as far as I'm going to go. I'm going to put this on your notes so that you can complete the rest of this strand yourself. So let's talk first about the enzymes that are involved in DNA replication. There are a number of different molecules and enzymes that are involved. You're responsible for knowing about 4 for the AP test. The first one is DNA helicase. I have a molecular diagram of the protein here as well. Remember, enzyme names all end in ACE. So that means this is an enzyme. So helicase is what unzips or separates the DNA double helix. As you can see in this diagram on the right, here we see the action of helicase. It is going to unwind the two strands of DNA, and it breaks apart the hydrogen bonds between the complementary nitrogen bases, allowing the molecules that are going to come next, the following enzymes, to have access to the nitrogen bases inside of the DNA molecule. The next enzyme that's involved is called topoisomerase. And topoisomerase is important because when you're unzipping a DNA molecule, if you think about if you've ever picked apart thread or strands of a rope, when you start separating them, things can kind of super coil up behind that, and that creates some tension in the DNA molecule. So to prevent that, you have this molecule called topoisomerase that helps relieve the strain of what we call supercoiling. So here you see this is a bacterial chromosome. When it is supercoiled, it's going to be twisted together a little extra, which is going to produce extra tension that could cause breaks in the DNA. So instead of having some unknown, unpredictable breaks in the DNA, topoisomerase comes in, introduces a break in the sugar phosphate backbone, lets the DNA kind of untwist, And then you can have another molecule, which we'll talk about in a moment, come back and fix any breaks in the sugar phosphate backbone and relieve some strain. DNA polymerase is a molecule that is going to actually be synthesizing the new strand of DNA. So what it does is it adds new nucleotides to the 3' end of a strand. So DNA polymerase, and this is going to become important later, must attach to existing existing nucleotides. So here we see kind of the molecular structure just in a general shape of DNA polymerase. This is the important part where you have an incoming nucleotide triphosphate, so in this case, an ATP. What's going to happen is this is the 3' end with that OH. The 5' end of that ATP nucleotide is going to come in. 2 of the phosphates are going to be removed, and then that nucleotide is going to be added into the 3' end. So DNA polymerase is responsible for synthesizing DNA, and the reason why DNA can only be produced in the 5' to 3' direction is because DNA polymerase can only attach to the 3' end of an existing sequence of nucleotides. The last enzyme that you're responsible for knowing about is called ligase. It catalyzes the formation of new bonds in a sugar phosphate backbone. So let's say this is a DNA strand and this is another sequence of DNA. You've got a hanging 5' end, a hanging 3' end, and ligase is going to come in and it is going to bond those two stretches of DNA so that there are no breaks in the sugar phosphate backbone. This is going to become important when we talk about Okazaki fragments in a little bit. And here's another, I like to include when possible, molecular diagrams of the molecules that are involved. This is ligase right here. And now let's talk about the actual steps of DNA replication. So what happens first is that helicase is going to separate DNA strands. At the same time, topoisomerase is going to help prevent any supercoiling. Then the next step is going to be the addition of RNA primers, which I'm going to talk about and have diagrams for each of these steps in a few moments. The third step is going to be DNA polymerase attaching to the primer and adding in complementary DNA nucleotides. This is going to happen in the 5' to 3' direction. And then any breaks in the sugar phosphate backbone that are present are going to be fixed by ligase. And we'll talk about why there are breaks in the sugar phosphate backbone in a few slides when we talk about lagging strands. So here's the first step. You have your 2 strands of DNA separated. The reason why you need to add what are called RNA primers is because DNA polymerase can only attach new nucleotides to an existing nucleotide sequence. If there's nothing there, DNA polymerase physically can't attach. So a different enzyme that you don't need to learn about is going to come in, put in a very quick, very small segment of RNA nucleotides called a primer. That primer is going to be there so that way DNA polymerase has something to attach to, something to add nucleotides to, and then later on that primer will be removed and replaced with DNA nucleotides instead. Once you have that primer there, DNA polymerase can come in, it attaches to the primer and starts adding in complementary DNA nucleotides. Notice it only works in the 5' to 3' direction. So this is going to become important in a few slides, which we'll talk about. Last of all, any breaks in the sugar phosphate backbone are fixed by ligase, and that's going to be primarily on what's called the lagging strand. Now, I've mentioned the lagging strand before. Let's talk about it here. So DNA is only able to be synthesized from 5' to 3'. That means as a DNA molecule is unspooled or unzipped, one of those strands, you're going to be able to synthesize DNA as long as the helicase is unzipping it. So you'll be able to continue synthesizing DNA. That's continuous replication, meaning it can happen without any breaks. And that strand that can have continuous replication is called the leading strand because it can just be synthesized straight through. However, for the other strand, it's running in the 3' to 5' direction. But that's not the way that DNA polymerase can work. So instead of having a brand new polymerase that's able to actually synthesize this way, what happens is that the helicase will unzip a segment of DNA, and as soon as enough space has been cleared on this lagging strand, which is what the other strand is called. A primer will be added. That's what that dark blue segment is. And then DNA polymerase will come in and synthesize a small chunk of DNA in the 5' to 3' direction. Then it's got to wait for more of that DNA to be exposed, and it can synthesize another chunk in the 5' to 3' direction. This is called discontinuous replication because it happens in small pieces. So a little bit of section of the strand can be synthesized at a time. That's also why this is called the lagging strand. It lags behind because it has to wait until a new segment of DNA on that strand has been exposed. And these little segments that are produced are called Okazaki fragments. And these Okazaki fragments, if you did not have ligase come in and join these breaks in the sugar phosphate backbone, the next time that DNA molecule separated into the two strands, those Okazaki fragments would float off. So that's why ligase is such an important molecule. It makes sure that you have one unbroken molecule of DNA. All right, that's it. Next up, we're going to start talking about protein synthesis. So if you remember codons, this is your jam.